Okay, tell me about,
is it on?
what do you notice about most individuals' DIS-80 genotypes? DIS-80 you will know all about on your take-home exam. That is the locus that we fingerprinted you. What do you just quickly notice about the class and the genotypes?
Most people have 2 bands.
So what does that mean from a genotype point of view? We've talked about this.
That there are 2 genes that are...
2 genes?
No, 2 fragments that
2
are...
genes? What does this mean? Come on, we've talked about this. You had it in discussion yesterday. Are these people homozygous or heterozygous?
Heterozygous.
In a sense.
Because
so
when
they have 2 genes or 2 alleles?
2 alleles? Because it's like the 2 different fragments, and so they're being on the gel.
So forget fragments right now. You're looking at genes, in a sense, DNA in your genomes. So don't worry about fragments. What do you see there, Tiger? Help Rose out. Alleles. What do you notice about, this is your DNA fingerprints. This isn't from a book. What do you notice?
Well, all of us would have the same genes, so they can't represent different genes, so it must be different alleles.
Yeah, so that represents very good. So there's a lot of different alleles of this particular locus in this class and at Davis. You notice that? Do you see that, Dora and Faria? But what about individuals? How many alleles do each of the individuals have for the most part, Tiger?
2.
Yeah, most of you are, let's say, heterozygous for this particular locus, which is what one would expect because we're basically hybrids, right? You get 1 chromosome from your mother and 1 chromosome from your father, and it's very unlikely that the allele is going to match in terms of exactly what the fragment is. DIS-80, as you will see on your take-home, is a VNTR. It has a basic repeat unit of 16 base pairs. And this is one of the VNTR loci, variable number of tandem repeats, which we're going to talk a little bit about today, and you know a little bit about. That's used for commonly in forensic testing. Do you have a question, Samantha? Okay, quickly.
I actually just wanted to ask what was being tested necessarily, because I see I'm like the
What is being tested?
Yeah.
Nothing's being tested.
What's the gene that we were looking at?
We're not looking at a gene. We're looking at a VNTR. You had that in discussion yesterday, variable number tandem repeats.
Okay.
So it's not a gene. It's just a sequence. Remember, as you'll see today, only about 1.5% of your genome represents coding sequences. Most of your genome represents all kinds of other stuff. So you're looking at a particular DNA sequence. Go ahead, Mike.
Why are there 2 samples in the well?
Oh, I don't know. You're going to figure all that out because there's 4 pages on your take-home that deal with this particular figure. Okay. So anyways, this will be the first and last time you're ever going to see, this is going to be the first, hold on. This is going to be the first and last time you're going to see your own DNA fingerprint. So there you are. And, oh man, I shouldn't tell this story. No. Once upon a time, I decided to fingerprint a family, an extended family of people that had a number of offspring. And so we got all kinds of samples. And we fingerprinted those samples. But I never showed the family the results because there were 3 different fathers.
Oh my God.
And those are the things that you learn when you do these kinds of things. And that's what we talked about a couple of weeks ago with people sending in all of their DNA and getting back all of these results. They find the hidden sequence in their family that they never knew about. And it becomes a very serious issue. Like, hey, I never told you that I wanted you so badly that I adopted you. That becomes a real big problem. Or, hey, I never told you that I got pregnant when I was in college and I had you, but the guy just drifted away and I found this other wonderful guy and he became, you know, your dad. I mean, it's those kinds of things that are going to, you know, come up in the course of these kinds of things. As you see, those pictures up there don't lie. Those of you that have 2 bands that are different, one comes from your mother and one comes from your father. I don't know which one's which. We'd have to, you know, do that with your parents to find out. And those of you who are homozygous, have same band size, one, of course, from your mother, one from your father, but around about the same size. Anyways, this is really neat stuff. But what you're seeing here is, in a sense, what people do, you know, police when they take swabs or any kind of DNA fingerprinting for parentage or any of these things, that's what you see. And it's really neat. And you saw how simple it was. It wasn't so simple in the lab. I mean, it took a reasonable amount of time, but it's not that complicated technically if you know how to do it and you have the right reagents and everything you know. Once upon a time, I did this class like this with Kyoto University in Japan. And I had about 40 kids in Japan, and I had about 40 kids at UCLA. And in that exercise, which was 3 years ago, I paid for and sent half of my class to Japan for a week. And then half the Japanese class came to UCLA. So it was a lot of fun. Here, because of the small numbers at Davis, John and I had originally planned to have some of you go up to Davis and half the class at Davis, but because of the small size, we're just bringing everyone down to Davis, and it's going to work. In any case, that's what's going on next week. Now, and today's lecture, okay, that's what we're going to do. We're going to finish factor 8, and then I'm going to go into genomics. So let me pick up where I left off last time. So this is still lecture number 4. And I think I can pick it up where I left off. The wonders of PowerPoint. Okay, as you recall last time on Tuesday I was talking about this wonderful quest to find a drug for factor 8 and to help find a treatment, not a cure, for hemophilia. And as I said on Tuesday, it was a lethal disease. And I said that they didn't know where the gene was expressed, but they knew a little bit about the protein, and they knew that the factor 8 was a blood clotting protein, and they had purified some of it from many, 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 many liters of blood, and also purified a comparable protein from about 20,000 liters of pig's blood and 20,000 liters of cow's blood, and they're able to get some of the protein, that is the factor 8 protein from humans, factor 8 protein from cows, factor 8 protein from pigs, and they're able to do a little bit of protein sequencing. And remember that the sequence of a protein starts at this N end and C end and has polarity, and there's a very specific sequence of amino acids in between. And so what was known then at that particular time was the sequence of the protein, but just a little bit of it. There we go. Just a tiny bit of purified factor 8 protein. And what was also known at the time, obviously, and you guys said that too, was that if you have a gene, even if you don't know where the gene is active, you know that the gene is in your chromosomes. You know that the gene is in the cells of all of your chromosomes. So therefore, if you want to find the gene, you have to make a library of the human genome and hunt for that gene, because you can't start with a copy DNA library, a library of the messenger RNA, because in the case of insulin in your discussion, you had to have known that insulin was active in the pancreas in order to find the insulin messenger RNA. So we've got to be able to make a library, and we've got to be able to eventually purify the gene in some way and move from there. And so what that means is that instead of going from the gene to the protein, as you did in your BLAST analysis in that quiz a few weeks ago, in other words, you knew the gene sequence and you put it into a database and it told you what the protein was. In this case, you have to go from the protein to the gene. And because of the genetic code, you can do that. We talked about that on Tuesday. If you know the genetic code, you should be able to predict the DNA or RNA sequence, although because of the degeneracy of the code, there's a little bit of a problem. So we've got to be able to start with the genome library. Now, in order to make any library, which is the collection of sequences in something that you want to clone, so for example, if you make a genome library, a human genome library, the library contains the collection of sequences in your chromosomes. And when the human genome was sequenced, They started with a whole big collection of clones of your DNA sequences. Everything starts with a clone. If you want to clone the pancreas messenger RNA and you want to have a library of pancreas messenger RNA sequences, you clone the pancreas messenger RNA and you have a collection of clones representing every different messenger RNA in the pancreas. Now, in order to do that, you have to have a vector. Remember that the vector is the vehicle which allows the DNA fragment, whether it's a cDNA or a foreign genomic DNA fragment, to replicate inside of a cell. You remember that from the first and second weeks of class. It's a vehicle. It's like a little chromosome which is able to replicate on its own because the vector has certain characteristics, Derek. It has a selectable marker, which allows you to select for its presence and distinguish it from its absence. It has an origin of replication, which allows it to replicate inside of a cell. It has cloning sites, meaning restriction sites in which you can add foreign DNA fragments or cDNA fragments. And it has, a variety of different characteristics that allow it to get inside of a cell, so you can clone in a test tube and put it back in, and get it outside of a cell. And so there's a wonderful collection of characteristics that are common for any vector. The vector that you've heard about so far are these two vectors. You've heard of two vectors, believe it or not. You've heard of this regular bacterial plasmid, which you used for many of your quiz questions, and we discussed with respect to the Cohn and Boyer experiment the first week. And we talked about the TI plasmid, the vector that was used to put plant DNA in, or put foreign DNA into plant cells. That's also a vector. Different kind of vector than a plasmid vector that we were talking about the first week, but it's still a bacterial plasmid vector, does different things, but it's a vector. Now, the vector that I want to focus on here in terms of making classical human genome libraries. Katie, no one does this anymore. There's probably only 2 people in this room. I know there's only 2 people in this room that have ever done this. And that's Dr. Harada and myself. Okay? No one does this anymore. But it illustrates how one makes a genome library, because regardless of whether you use this vector or not, in order to sequence the human genome, you had to clone it in some vector. It wasn't this vector, but it was a vector that had similar kinds of properties. So in this particular case, to clone the human genome, you have a big problem. The problem is the genome's big, really big. And so that means that when you make a library of the human genome, if it's really big, you need a lot of clones. And that means that you might have tens or hundreds of thousands of clones, and that's really hard to handle. So the general rule is that the bigger the genome that you want to make a library, you want to use a vector that can accommodate very large DNA fragments. And generally, normal bacterial plasmids handle DNA fragments that are relatively small. And so that means if you have a vector that only takes small DNA fragments, when you want to clone the whole genome, you need millions of clones in order to cover every DNA sequence in the genome. If you use a plasmid that takes enormously large DNA fragments, it means that you have fewer numbers of clones in which you have all the DNA sequences in the human genome. Eden, would you rather handle in the lab a library that had 100 million clones or 100,000 clones?
100,000?
Yeah, absolutely. Because to search through 100,000 clones for your favorite gene is a heck of a lot easier than searching through 100 million clones. Remember, you have to look clone by clone by clone by clone in order to find the one you want. And it just so happens that the vector that's used to that was used to clone the human DNA to sequence the human genome were these vectors here. And I'm not going to go into them, but they're actually artificial chromosomes that took huge inserts of 300,000 base pairs or longer. So the idea was that you were working with a smaller number of clones because you have to handle it in the laboratory. Anna, is that clear? You sure? Remember those little colonies that you had on your plates, or your little schmears there? How would you like to have handled 1 million of those plates? Or would you rather handle 1,000? 1,000's a lot easier. So the deal is, again, if the genome is big like the human genome, which is 3.2 times 10 to the 9th base pairs, and you want to clone every DNA sequence in that genome, you want to put the largest DNA fragment you can in your vector so that your library has the fewest number of different clones, because just like a library that has books in it, I'd rather go to a library that has 10 books to find my book than to go to a library that has 10 million books to find my book. It's a lot less work. Is that clear, Julie?
That's clear.
Yeah. I don't want to dwell on this too much, but that's the concept. Go ahead. Go ahead. Shoot.
Now that we have the database that has the whole genome, it doesn't matter if we have 10 million or
Yeah.
100 million?
Good question. Now that we have the database, you don't clone anymore. You don't clone the human genome anymore unless I want to know about your genome. So then I got to make a library of your DNA. But we already have the sequence of the human genome, but it was a collection of individuals. You know, about 10 individuals donated their DNA and it was sequenced all over the world. But if I want to know what your DNA looks like from a sequence point of view, I still got to make a library of your individual DNA. But we do it in a slightly different, much more high-tech way, and I don't want to get into that. I debated about whether I was going to get into it or not, but I decided not to. Okay? This is historical. Okay? This is where it all begins. You can't get any DNA sequence unless you have a clone. You can't get a clone unless you have a library. So in order to clone the human genome and most eukaryotic genomes, historically, we use viruses. Viruses that infect bacteria. And I talked a little bit about these viruses because these are the viruses that the restriction enzymes fight against. Remember, restriction enzymes are present inside bacteria. And remember I said that they're used to defend the bacteria against invasion of foreign DNA, and they cut the foreign DNA into little fragments and destroy it. Well, these are the bacterial viruses in which the restriction enzymes fight against, but I'm not going to get into that story either. The point here is that these bacteriophages, you can assemble them in a test tube, as I'll show you, and you can use their chromosome, their actual viral chromosome, to stick a foreign DNA into. And therefore, you can use the virus, as a vehicle to get the foreign DNA inside a bacterial cell using a natural infection process. That's very different than transformation. Transformation, in the way in which we use it for genetic engineering, is an artificial process. You isolate the plasmid using these centrifuges like you had the first week of class. You then do all your restriction enzymes and ligations and all those things, and then you put it back into a bacteria using, calcium chloride and all kinds of chemicals to get it back in. It's not natural. When you clone something in a bacterial virus, you're able to get it back into a bacteria and make a clone through a natural infection process. Again, the little bacterial plasmids that we talked about accommodate small pieces of DNA. 1,000, 2,000, 3,000, 4,000 base pairs. As you'll see, the viruses, the chromosome of the virus, can accommodate up to, let's just say, 20 to 25,000 base pairs. So the advantage of the virus as a vector, as you will see, is twofold. One, you can accommodate a larger piece of DNA, which means you need a smaller library. Smaller library means it's easier, Max, to sort through and find the sequence you want. And secondly, the process of getting the foreign DNA in the bacterial cell, Anna, is very efficient. It's natural. The virus, spent 3 billion years evolving to do that. And so it goes in very efficiently. The plasmid is very inefficient. And because of that inefficient process, it's not very good for making a large number of clones. But the virus can make a large number of clones because it goes in really efficiently. I'll tell you what that means a little bit more in a second. Okay, so let me tell you about this virus. It's called Lambda. It's a particular kind of bacterial virus, but it is a vector. It can be used as a vector. The structure of the bacterial virus is like this. It has a head, it has a tail, and it has these little fibers. The head is where the DNA goes. The tail is what attaches to the head, and the fibers are what attach to the outside of the bacteria. It's a really elaborate little machine, and it can stick around in a sewer for literally billions of years. It's made up of proteins. There's a number of proteins that make it up. There's proteins that make the head, there's proteins that make the tail, and there's proteins that make the fiber. And what's neat about this virus is if you isolate the proteins from the head, you isolate the proteins from the tail, and you isolate the proteins, that are at the end, the little fiber proteins, and you put them in a test tube, and you add naked DNA, it spontaneously forms the virus. So you can make the virus in a test tube because there's no enzymes, there's no energy. It's an automatic, spontaneous process of reconstruction. So that means if you have viral DNA, pure, and you have each of the proteins and you mix them all together, the virus spontaneously forms. And that makes it very convenient for using it as a vector for cloning DNA. It's really neat. And furthermore, when you reconstitute the virus, Julie, it's infectious. And you can do this with a lot of bacterial viruses. You can do this with a lot of plant viruses. Can't do it so much for human viruses because they're a little bit more complicated, but you can do it. Now, look at the genome. This was one of the first genomes ever sequenced way back when using the sequencing procedure you used in discussion. Remember with the little, it was called Maxim Gilbert, and it was in the article that you read the second week of discussion. And that was used to actually sequence this genome way back when. And it took a long, long time to do it. Today we could do it in about 2 minutes. It took a few years for them to do it 25 years ago. But the fact is it's 49,000 kbs. A kb is 1,000 base pairs. So it's 49,000 base pairs in length. And it has all the essential genes. So notice it has genes for the head, genes for the tail, and it has these genes called lytic functions. And what the lytic functions are functions that allow the virus to replicate and destroy the bacteria. This is, in fact, a killer virus. It survives by destroying the bacteria that it infects. And I'll show you that in a second. And so what we have here is the head, the tail, the replication functions, and then we have this thing in the middle. And I'm not going to go into what this thing in the middle is. It has some genes, but they're not essential for the life cycle of the virus. They can be done away with. And this is where you can clone a foreign piece of DNA. You can take this replaceable region out by using the relevant restriction enzymes. You can isolate what we call the left arm and the right arm. You can just isolate them in the test tube. You can then anneal it with foreign DNA, as you'll see. And you get a new chromosome, a chimeric chromosome, that would have this arm, and a foreign DNA in the middle. And then you can package that into the head of the virus. And now you have a recombinant virus that now has viral DNA plus foreign DNA, and it behaves perfectly normally, doesn't do anything, recognizes it as its own, can infect bacteria, and it goes through its life cycle in a really neat way. There is the genome of the virus. How does this work? There's the life cycle. Virus attaches to the bacterial cell wall. It has receptors. It knows what bacteria to bind to. And I should say the virus is species specific. The Lambda virus will only infect E. coli. It's not going to infect Salmonella. It's not going to infect Shigella. It's not going to infect some strange, Vibrio cholera. It's specific for E. coli. This is an E. coli bacteriophage. How many of you people remember this name Max Delbruck from the double helix? Julie? Max, remember? James Watson said, yeah, Max Delbruck, you're highly recommended, Max Delbruck. The name. He's the father of this science. He won a Nobel Prize for being able to say that we should use viruses as a simple form of studying genes. This guy was a real luminary in molecular biology in the 40s and 50s, 60s, 70s, and he passed away in the 80s. But the point here is that, again, someone had to isolate these viruses. Max was one of the first people that did that, study its life cycle, and he said, hey, this is really a novel little thing that we can use to study genes because it doesn't have a lot of genes. But in this particular case, the virus attaches to the E. coli. It actually injects its DNA. It does. There's a very famous experiment that demonstrated that, which I unfortunately don't have time to get into, but it's a lot of fun to talk about. The DNA gets in. It's called the Hershey-Chase experiment. The DNA gets in. Daisy knows all about that. The DNA gets in, and when the DNA gets in, it specifies some proteins, and those proteins that it specifies in its genome take over the E. coli, take over all the apparatuses of the E. coli, take over its protein synthesis apparatus, take over its DNA replication apparatus, starts replicating the viral genome, starts making viral proteins, self-assembles inside the cell, and it makes an enzyme that destroys the bacteria. And the bacteria is destroyed, viruses are thrown out. Remember, this is happening in a sewer. So viruses infect one E. coli, they replicate, bursts. There's a lot of other E. coli in the sewer, infects other E. coli, et cetera, et cetera, et cetera. Goes on like a sort of a chain reaction. Now, when you do this in a lab, you do it on a plate, exactly like the plate that you guys had, except you have what's called a lawn of bacteria, meaning the bacteria is evenly spread on the plate. And you infect the bacteria with the virus, and the virus generally, one virus goes into one bacteria, in a sense. It replicates, and when it replicates, it destroys a number of cells around it because it infects one cell, bursts, infects the next cell, bursts, infects the next cell, bursts, and infects the next cell, and it makes what is called a plaque. As you saw on your plates, remember your plates, Dora? They were kind of hazy. Remember that stuff looked kind of yellowish and stuff? Well, what the virus would do would destroy that. So if the virus infected those plates, you'd look down and you'd see it clear. You'd see it go from that hazy to a very clear look. And that clear look is called a plaque. There's the virus, here's the bacteria, and in the middle is the virus. And that's where the virus is. And then furthermore, one virus, one bacteria, and essentially you're getting a clone of viruses in a plaque. That means each plaque, when you use it as a cloning vector, the virus is one foreign DNA fragment. So in this case, if we were cloning the human genome, each plaque here would represent a different human DNA sequence. So you'd have human DNA sequence 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, et cetera, et cetera, et cetera. And to get a library of the human genome, of course, you need 100,000 of those or more. But the point here is that this virus can be used as a vector. And its selectable marker is not antibiotic resistance because it doesn't have any antibiotics antibiotics resistance genes in its genome. Its selectable marker is killing the bacteria. That's its marker. It kills the bacteria, forms a plaque. You know it's there. That's the selectable marker. It's analogous to an antibiotic, but it's not. OK, so what are the advantages of this particular vector historically? Well, I think I said it. It can accommodate a huge piece of DNA compared to a normal garden variety plasmid. It can infect these cells naturally. It's a very efficient process, and that means you can get lots and lots and lots of clones relatively easily, and finally you can make this virus inside a test tube. We used to spend a lot of time in the old days doing this, but this is, again, 20 years ago. So how does this work? Well, the first thing you have to do when you're doing this is you have to isolate the virus from the bacteria, Lauren. And you have to isolate the viral DNA from the bacteria. And if you know the sequence of the viral genome, you know what the restriction enzyme sites are because the sequence predicts the sites on the genome. Remember that a genome has a particular sequence to it. There's a particular sequence to the genome. Remember that that sequence goes 5' to 3', and that sequence consists of sequences that recognize restriction sites, and therefore you have a certain number of restriction sites along the genome representing different restriction enzymes. And if you know that, And if you know what that is, and you know that there's a site here and a site there, and this is the right arm, let's just say, and this is the left arm here, you can then purify this arm and purify that arm and get rid of this replaceable region. Does that make sense? And you'll know all about how to make a restriction map because it's on your take-home test. And so the point here is if you understand the DNA sequence, you have a restriction map, and you know from your quiz from last week that unless you know where the restriction sites are, you can't do any genetic engineering. Genetic engineering depends upon a knowledge of where specific restriction sites are. So if you want to add a switch to a cDNA, you've got to have appropriate restriction sites. If you want to add a regulatory sequence to a switch and combine them together with a particular coding sequence, you have to have specific restriction sites in order to do that. So the garden variety, well, I shouldn't say garden variety, the main tool of a genetic engineer, the main tool is restriction enzymes in the old days, and sites, because that's the way you put things together, like your Lego kit. So you have to have a knowledge of those sites. So in this particular case, let's say we know what those sites are, and we know that there's a restriction site right here and a restriction site right there, and you can see it. It's for EchoR1, and you can see that you can separate this arm, and you can throw away the thing in the middle. And you can separate them. I'm not going to go into how, but you can separate them. So you can get a vial of the arms of the viral genome, left and right arm, and those are the arms that contain the essential genes that are necessary for viral replication. Then you can take your human DNA that you've isolated from whatever source you want. You can digest it with the same restriction enzyme. You can then anneal them. They have the sticky ends, the complementary bases. You can anneal them together. You can then use a ligase to seal the 3', 5' phosphodiester bonds, and you get a hybrid virus. Now the virus has a DNA fragment from your genome, and it has its essential genes that's required for DNA replication. Now, in your same lab, you have head proteins, you have tail proteins, you have fiber proteins that you've purified from purifying the virus. You put them all together, and this recombines into a virus. And now you just take that virus, you infect the bacteria. There they are. It injects the chimeric DNA into the bacteria. One virus per one bacteria. That means one foreign DNA per one bacterial cell. They make plaques. It means one plaque per one human genome DNA fragment. If you need 100,000 to get a whole library, you just collect separately 100,000 plaques and you make a library. Stephen, is that clear? It's pretty simple. That's how it works. So what you would do is you would get all these plaques. Each plaque represents a different human gene. And then you can just mix them all together and you make your library. And then you can rescreen them, do anything you want. We still have libraries in my freezer that we've had for 25 years and they're still good. Once you make them, they're there. Back to your question, Julie. If you have the sequence of the human genome, do you need to do this? No. But what if you need a particular gene? Well, the modern way we would do it is if you have the DNA sequence, you use PCR and you just take some human DNA and use a polymerase chain reaction. You know what the sequence is and you get the fragment that you want. But you still may have to do some cloning, but it's a little bit easier. But again, that took 25 years to do. Okay, now, here the going gets a little tough. So I want you to listen to me very carefully. Jessica, am I making myself clear? Samantha, am I clear? Brian? Clear? Good. But now I need you to listen, because this is very important. Brian, what's an enzyme?
It's a protein that has
Good.
some specific function.
It's a protein. Great. What's that protein? What's the enzyme do, Dora? What's the function of an enzyme?
I think it has different enzymes have different functions.
Yeah, but what does it do in general? There's a property of all enzymes. What does it do?
Oh, it cuts?
Well, that's a restriction enzyme. But in general, what does an enzyme do? Faria, what does an enzyme do? Think about your little chemistry from high school. Eden,
Oh,
go ahead. Tell Faria.
It's a catalyst to a reaction.
It's a catalyst, exactly. It's a catalyst. And it's a catalyst that allows that kind of reaction to go. Is that correct? In the case of a restriction enzyme, what reaction is it carrying out?
Cleaving DNA.
Yeah, it's cleaving what bond though is it breaking?
The phosphodiester.
Yeah, so the restriction enzyme is a catalyst that's digesting and breaking a 3' to 5' or 5' to 3' phosphodiester bond. Okay, Eden, I'll pursue this. This reaction, and I'm not going to get fancy here, does that reaction go with a certain speed?
Yes.
Yeah, each reaction has a particular speed. Do you agree, Lauren? Okay. If I added a lot of enzyme or a little bit of enzyme, might the reaction change? Yeah. So if I add just a little pinch of the enzyme, that reaction might go really slowly. If I add a ton of the enzyme, it just might go like gangbusters. And I'm not going to get into this because those of you that are going to go on in science and biochemistry are going to have your full of this. But the point here is that these reactions occur at a certain speed. And you can adjust that speed. Now, why am I telling you about this? Because I know those of you who are majoring in comm studies don't really care about this very much and probably won't remember it, but it's very important for understanding how the human genome was sequenced, and it's very important for understanding how you make a library. Let's say, Eden, can I pursue you? Let's say we have in the human genome, do we have one EcoR1 site?
No.
How many do you think we have? Thousands?
4,000?
Well, tens of thousands.
Tens of thousands.
Yeah, we have tens of thousands of sites. Okay, great. There's a site, there's a site, there's a site, there's a site. Let's say this is a chromosome, and I have gene A, I have gene B, I have gene C, I have gene D, I have gene E. Now, what happens if I digest that DNA very quickly? How many fragments will I get?
5.
Okay, I'm going to get fragment A, right? I'm going to get B, right? I'm going to get C, right? I'm going to have D, right? I'm going to have E, correct, in simple form. Now, how many clones can I get from that, Julie? How many clones can I get? Look at that diagram.
We can get 5 different types.
Are you sure? You're positive. Dora,
No,
think
you can
about
get
this.
5
I can get 5. I don't
types.
see any restriction sites on the ends. How's that going to clone?
Oh, 3.
You have to have restriction sites on both ends of the clone, right? So in this case, I can clone this one, I can clone that one, I can clone that one using that enzyme. But I don't have an EcoR1 site here. It doesn't clone. Is that correct?
Yeah.
Okay, just a technical, minor technical thing. Now, in my library, whether I use the virus or whether I use the plasmid, let's just ignore the outsides for the time being. Let's just include them. Let's say that there's sites on the outside, just for the sake of simplicity. Got a site here, and I got a site here. So now I have 1, 2, 3, 4, 5 fragments. How many clones, Julie?
5.
Yeah, how many, Eden? You agree? Okay, and they're in this library, right? And let's say they make 5 different plaques. You with me, Maura? We've mixed them together in a test tube, right? Where did they come from originally? Use the microphone. Where did those DNA fragments come from originally?
Human.
Where?
The
Where are they in your chromosomes?
chromosomes.
Yeah, originally they're in your chromosomes. And let's say they were all next to each other, as you can see. But you didn't know they were next to each other, did you? Because you don't know anything about it, right? But now they're in this test tube. A, B, C, D, E. Is there any way you can show the relationship between any of these fragments from this? Are they completely independent of each other?
Yes.
Yes, what?
They are.
They're independent of each other.
Yeah.
They're all independent of each other. So you would never be able to know from this that A is next to B and B is next to C and C is next to D in the genome just from this. Is that correct?
Yes.
It's exactly correct.
Well, then also.
No, wait, let me finish. What happens if also we have these 5 restriction sites, and let's say that's one gene. And let's say that's part 1, part 2, part 3, part 4, part 5 of that gene. And now we've done this enzymatic reaction. We have 5 fragments. What did you do to the gene, Tiger?
You've destroyed it.
You've destroyed the gene, exactly. And they'd be in your library, and you'd never know that they were actually part of one gene. Because you don't know anything about the gene before you clone it and study it, right? It's not like Julie says we have the sequence of the human genome, right? So you'd be pulling out these fragments, and you wouldn't know their relationship. You'd just know that they were separate DNA fragments, and you wouldn't know anything about them. OK, Lauren. What happens if I go back to my enzymatic reaction here? Same piece of DNA. OK, let's go back to the A, B, C, and D. A, B, C, D, E. That's in the chromosome inside the genome. A, B, C, D, E. Those are genes along the arm of that chromosome, okay? Now, what happens when I carry out this reaction, A to B? In this case, I'm taking a double-stranded DNA molecule. I'm cleaving the phosphodiester bonds, and I'm getting, let's say, 2 double-stranded DNA molecules, in a sense. Now, what happens if I slow that reaction down to a really, really, really slow rate, and I time it? Do you think that each of these restriction sites are going to be cleaved at exactly the same time?
No.
And what if I have a population? Remember, I'm not working with one molecule when you do these things. You're working with millions of the same molecules. Meaning, in my test tube, I'm not going to simply have one of these pieces of DNA. I'm going to have one like that. And I'm going to have another one that's identical to that. Remember, a mole of DNA has 6 times 10 to the 23rd DNA molecules. You all remember Avogadro's number from your chemistry. So we don't deal with moles of DNA, but we deal with, 6 times 10 to the 10th molecules when we do this. So now I'm slowing it down, Lauren. Are all these restriction sites going to, if I have a huge amount of enzyme, they're just going to be blasted away. Agreed?
Yeah.
But now I've slowed it down. You with me, Dora? It's going real slow. What do you think is going to happen? Are these sites going to be digested independently? It's like walking the perimeter 1 inch an hour.
They're going to be digested independently.
Yeah. So in this molecule, you might digest this at that time period, right? And in this molecule, you might digest this in that time period. And in this molecule, you might digest that in that time period. And so what you're going to wind up with is a fragment here and a fragment there. You're going to wind up with a fragment here and a fragment here. And you're going to wind up with this. Do you notice anything about that, Thomas? Notice anything interesting? And remember, when you clone this, this would be one clone, that would be another clone, that would be a third clone, that would be a fourth clone, that would be a fifth clone. They all go into different vectors. Do you notice anything interesting about this?
It seems pretty random. You can't control where the...
But do you notice any pattern there relative to A, B, C, D, and E genes?
They're each broken in separate places. So.
They're broken in separate places. What do you see about, I know it's not the nicest drawing in the world, but what do you see about this fragment that has A, B, C, and what do you notice about this fragment that only has A?
One's smaller, or they go up by 2s, technically.
Again, let's say one of them is like this, and the other one is like that. What is this called? There's a term for that. It starts with an O. What am I doing with my arms?
Overlapping.
Exactly. You get overlapping fragments. That means that this clone by itself is related to that clone by DNA sequence relationships. And this clone is attached to B, C, and D, or B and C, and this one's attached to A, B, C, D, and E. If you make your library like that, Lauren, Now can you find the relationship between these clones, and can you understand how these genes were next to each other in the genome? Taylor, do you see that conceptually? In other words, if you digest your DNA all the way, you get hundreds of thousands of individual DNA fragments, and you can't establish the relationship between them. But if you slow the reaction down and get longer fragments randomly overlapping each other, that means one clone is going to contain some genes that another clone has. Another clone is going to have some genes that another clone is going to have. That clone is going to have some genes that another clone has. And could you imagine if you had a chromosome, could you imagine just starting and going like this and finding a whole bunch of overlapping clones that go all the way down the chromosome in a sense? And if you knew this pattern, you could start here and end up here in a very ordered way. And you'd know that this clone was at this end and that clone was at that end, and you'd get all the clones in between. When you make a library of the human genome for sequencing, even in 2008 or 2009, even today, Julie, you have to have what's called overlapping clones. You have to have DNA fragments or clones that overlap each other so that you can find out the relationship between the DNA in those clones with each other. Because remember, originally they were in a chromosome in the genome. In the genome, Jessica, these genes were next to each other on 24 pieces of chromosomes. 23 autosomes, 22 autosomes, an X and a Y. You have to find out how they're related to each other. If the gene is very big and you blow it apart, you'll never know that the gene is the gene. But in the case of slowing the reaction down, Lauren, you create a library, and in that library, each phage will contain maybe many genes. And these different phage, the foreign DNA fragments will overlap each other. And if they overlap each other, that means they have DNA sequence complementarity to each other. And that means you can go from one phage to the next phage to the next phage to the next phage, and you can find out their relationships and how they were originally organized inside the genome. This is essential. Julie, the human genome would have never, ever, ever been sequenced without doing something like this. Because all you would have had is a random collection of clones, and you would have never known how to put them all together. It's like if you had a jigsaw puzzle, and the jigsaw puzzle, you know, has all these little things, and you've got to find the ones that get together. Well, if they were all square, you'd never be able to figure it out. That's sort of the analogy. So in this case, the overlapping clones are the little things on the jigsaw puzzle that allow you to put things together to show the picture. And what you do to do this is you control the rate of this reaction. And so instead of, like you did in the Cohn article the first week of class, blasted that DNA to pieces, breaking up the genome in as many pieces as you want, you slow it down, Rose, just like your metabolism's been slowed right now. You slow it down, and you make big pieces, and those big pieces overlap each other. So let me conceptualize for you. We make a library of the human genome. In this case, we're searching for factor VIII. And here we go. Here's the virus. We take this arm and that arm, and we get rid of that yellow stuff in the middle, because we don't need that for the replication of the virus. We purify it. We digest the human DNA. It's called partially. So we do it just a little smidgen, and we get these series of overlapping DNA fragments. One of these fragments gets joined with the two arms. Let's say they were digested with EcoR1. We ligate together after annealing the sticky ends or complementary bases at the end of the restriction site. We ligate them, and we make a recombinant phage. We package them in the phage because it self-assembles, as I've told you. We infect our bacteria, we get plaques, and we make a library. But now we have a library of fragments in which we can have one clone, in which we can find another clone which has some overlap to it, so we can know what its relationship is in the genome. I know that was all a mouthful. but that's how it works. Okay, we already did that. So I'm not going to go through this. You can read all of this. It looks pretty complicated, but please digest it because you're going to need it for your take-home test. But the point here is, simply conceptually, Lauren, if you add a whole bunch of restriction enzyme, it's called a complete digest, and you get pretty small DNA fragments. If you add just a smidgen, it's called a partial digest, and you get overlapping DNA fragments. And you can overlap all of those fragments so that you can use one clone to find its neighbors. And how in the heck are you ever going to know when you see these chromosomes and you see those genes on those chromosomes? You have to know the relationship between them. Samantha, do you have a question, or are you fixing your hair? Pardon? Fixing your hair. So here we go. Here's a piece of the human genome. Here are our overlapping DNA fragments. Notice that this fragment has part of that fragment, that, so on and so forth. And so I can use this, as you can see, in order to get an overlapping set of sequences that represents the entire human genome. Okay, let me go back. So that's how it's done. Is that clear, Marvin? So now, we made our genome library. We have these overlapping fragments. And now we got to find the factor VIII gene. How are we going to do that? We only know the protein sequence, so we have another problem. I want you to listen carefully on this one. Let's skip this stuff. So there's the sequence of the factor VIII protein. So we know this sequence right here, and we know the genetic code. And remember I told you, Harumi, that the genetic code is degenerate. That means there isn't one unique sequence specified by the amino sequence of the protein. Do you agree with that, Max? But you have the code in your hands, like translating a foreign language, right? So if I know the protein sequence, which is cis, luz, tyrosine, et cetera, I can predict what the codons are going to be. So in this case, we have 2 codons for this amino acid. In this case, we have 3, 2, 1, 2, 2, 2. How many different synthetic reactions am I going to have to make in order to get probes to find the factor VIII gene? What I mean is that in order to get the probe, you have to go from the protein sequence, you have to predict the nucleotide sequence, 5' to 3', and then you have to synthesize using chemistry in a lab, one nucleotide at a time, the exact sequence you want. Ryan, how many times have you had little oligonucleotides synthesized?
Several hundred.
Kelly, how many times have you had this done? It's pretty easy, isn't it? You don't do it. You send it away. You say, this is the sequence I want, and it comes back in the mail. But it's all done by chemistry. It's not done by PCR. It's not done by DNA polymerase. It's actual chemistry. They take the A, they join it to the C. They take the C, they join to the T. People can do this synthetically. So if you can do that, and you know what the codons are, you can predict how many probes you have to make. And when I say probes, I mean radioactive probes, meaning radioactivity so you can detect it. So here I have 2 possible sequences, 3 possible sequences, 2, 1, 2, 2, 2. And so therefore I need to make 48 different kinds of synthetic probes. Meaning, Anna, there's 48 different combinations of DNA sequences that are specified by that one protein sequence. And again, the concept is, Ben, that the code is degenerate. If the code wasn't degenerate, you could go right from the amino acid sequence to the nucleotide sequence, but you can't. You understand that, Kathy? Okay. So we make 48 synthetic probes, meaning one sequence will be the one you want. There's going to be 1 out of 48 that's going to be the exact gene sequence of factor VIII that specifies that protein. You make 48 probes. They're all radioactive. There's another way of looking at it. And then you do it synthetically, and now you plate out your library on these plates, and you do exactly what you read about in week 2 of the discussion. You take your probes, you take this little filter paper, you put it over all of these colonies. It takes a little part of each plaque which means each clone's DNA goes in its place on this little filter paper. It's kind of like, it's called in your article colony hybridization. In this case, it's plaque hybridization. You denature the DNA on the filter paper, and you mix it with these 48 radioactive probes. And because only complementary bases will form a double helix, all the others won't form, and there's only one out of the 48 that's going to find the factor VIII gene. And that means, let's say, one clone. You're going to be able to find, even though you have many plaques on this plate, you're going to find one that's going to form a signal by autoradiography. The protein sequence is very short, tiny. So you're only searching for a short segment of the factor VIII gene. Samantha, I can't hear you.
So just going back to the slide before, we were talking about, well, we have proteins and we're going backwards because we have the proteins and we know that these proteins are made by different amino acids. And you were talking about...
I didn't say anything about proteins being made by different amino acids. I said the codons.
Well, the codons, which lead to
So
the
what's
amino
your
acids.
question?
My question is how do we know if there's 64 different kinds of combinations?
You're going to figure that one out on your take-home test.
Oh, my. Okay.
It's pretty simple. And you'll study it when you look at these notes. It's not that difficult to do that. Just simple. OK, so the point conceptually is that you have these probes. Here they are. We have 48 of them. One of them is going to match a sequence of the factor VIII gene. Remember, we're annealing. We're getting a double helix. One strand radioactive. The other strand is in the phage. The radioactivity is going to be on this filter. The filter is going to be exposed to X-ray film, like you have X-rays. And exactly where it is on that filter, you're going to see it there. Then you go back to the plate. Be this one right over here. You purify it, and now you have the clone you want. Tiger, is that clear? Good. So they did that. That's what they did back in 1982 and 1983. They used this synthetic sequence that they got from many, many liters of blood, this very precious protein, which is very difficult to sequence. They only had a short piece of it. They used the genetic code. Without the genetic code, they couldn't have done this. Without the invention of recombinant DNA, they couldn't have done this. Without being able to use phage as vectors, they couldn't have done this. Without restriction enzymes, they couldn't have done this. With all the stuff that you've learned about. And lo and behold, they find that there's the clone that they got right over there. And they knew a little bit how to sequence in those days. And they sequenced this, and this is very important. By sequencing, they showed that the sequence on the clone matched exactly what they predicted from the sequence of the protein. That's critical in this experiment. You have to sequence what you get to show it's what you want. And then they sequenced this whole fragment that they got out of that. Remember, one fragment they got. With me, Julie? It's one clone. They sequenced the whole thing. They couldn't find the whole factor VIII gene. It was just a tiny little part. A lot of the DNA sequence was gibberish. They were just learning, oh, it had introns. And they're going, my God, what are we going to do in terms of getting the whole gene? And so what they did was something else. They knew that they made this overlapping library. So they knew that if they had this clone, they could use a particular procedure that I'm going to tell you about in a second to go from one clone to the next clone to the next clone to the next clone because they had overlapping DNA sequences. Anytime you have overlapping DNA sequence, you can use one DNA sequence to find the other. Denature, anneal, denature, anneal. It's all complementary bases, double helical structure. Clear? So what they did was that they took this clone and they went back into their library and they used this as a probe to screen for the things that had complementary sequences. And they found this clone and that clone. And they sequenced that and found out, well, we don't have the factor VIII gene. So they took this clone and went back into the library and found that clone, and they still didn't have the whole factor VIII gene. And they took that clone and went back into their library and they found this one. And all along the way they're sequencing. And remember what you know about genes. There's a start, there's an end, and they knew what those sequences were. So in this sequencing process, they were looking for the promoter, they were looking for the terminator, and they were looking for all the codons in between and everything else. So you have to sequence while you do this. And lo and behold, they found that the factor VIII gene was 186 nucleotides in length. Can you imagine that? The globin gene is 1,000 base pairs in length. This gene is 186,000. It's one of the largest genes in the human genome. It was bad luck for them. They didn't know that at the time. This took like 3 or 4 years to do. It was a really tour de force. But the point here is that what they found out was that the factor VIII gene is 186,000 nucleotides in length. They found out that the protein was 2,351 amino acids, which is huge. They found out that the cDNA was 9,000 nucleotides in length, that is, just putting the exons together, and it had 25 introns. You think that's an easy gene to figure out, Ben? 25 years ago with no gene bank? That's a tour de force. They're not going in any blast, I can assure you of that. They had to figure this all out on their own. Let me digress. In order to find this gene, they had to do this thing called walking. Remember, Lauren, that your library contains overlapping DNA fragments. If I take one fragment from your library and I make it radioactive and I rescreen the library, I'm going to find all the neighbors that overlap with that fragment. Is that clear? And because they overlap, I can march from one clone to the other in the library just by screening, getting, making radioactive, screening, getting, making radioactive, and I can do some certain things with it. So it's called walking, and that's what they did. So you have one clone, you take a piece of that clone, you rescreen the library, and you get an adjacent one. You find that one, you take a piece of that one, you rescreen the library, and you find adjacent ones. You keep doing it over and over and over again. And the library, Julie, to make the human genome, they overlapped 24 different chromosomes with clones.
24?
Yeah, X and Y. 22 autosomes, X and Y, and they overlapped them all from one end to the other. Thomas, how do I know that I'm overlapping? What is a marker for overlapping if I can't sequence?
Well, they would be complementary, so they would match.
I know, that's the one thing, but how would I know it physically if I did something?
The radioactive marker?
Yeah, but what else? Once I had them, I don't need the radioactivity anymore. What would tell me that they overlap? What does the sequence have contained within it? It's figure 2 of the second article that you read in your discussion on making recombinant proteins. It's the second page, bottom figure. What do DNA sequences have? What do you use to digest the DNA?
Restriction sites.
Yeah. So do you think that the restriction sites on these clones would overlap with each other?
Yeah.
That's your marker. So in order to find that out, you have to either sequence them, but in the old days we made restriction maps. And so there would be sites that overlap. There would be sites here that are different. There would be sites here that are different, but in the middle they overlap. And that's how you know you have overlapping clones. So you have to make restriction maps in order to do that. And so you can go and march from one end to the other. And that's what they did, Julie, when they first started to sequence the human genome. They made what was called a physical map of the human genome, meaning they made a restriction map of every single overlapping clone from every chromosome from one end to the other. And once you do that, then you know where they are in relationship to one another in order to sequence human genome. I'll go into that probably on Tuesday.